My name is Maxim Kuleshov. I am a bioinformatician at the Mayan Lab in the Department of Pharmacological Sciences at the Icahn School of Medicine at Mount Sinai. Today I'm going to demonstrate a new resource we developed called Gene and Drug Landing Page Aggregator. High-throughput methods for screening small molecules and generating omics data have created an abundance of data related to variants, genes, proteins, and drugs. Additionally, storing and making data publicly available in various databases and web applications has become increasingly inexpensive and straightforward. Many biomedical resources provide information about variants, genes, proteins, drugs, and small molecules as dedicated landing pages. Providing links to such landing pages all in one central place can enable researchers to better find and access gene and drug-centric knowledge. The Common Fund Data Ecosystem, CFDE, Gene and Drug Landing Page Aggregator, GDLPA, is designed to accomplish the same by gathering 52 gene and 16 drug resources and databases that provide landing pages into one website. The website has a simple search tool that only requires the user to enter either a gene, a variant, or a drug name. The landing page provides a search bar for querying gene symbols, drug names, or variants. A drop-down on the left from the search field can be used to switch between the gene, the drug, or the variant search type. The search results can be additionally filtered by whether a resource is an NIH Common Fund supported resource. Filtering can also be done by the source type, whether a resource is a primary source that generated the data or an aggregator of data from many other resources. When a user submits a variant ID to GDLPA, it makes a request to myvariant.info API to resolve this ID from various variant annotation databases. Then, GDLPA looks for variant annotation data from dbSNP and uses the most recent mapping of the variant ID to a gene name. Then this gene name is submitted back to GDLPA, not as a variant input, but as gene input type. After the variant is converted to a gene, GDLPA displays a message with this information. The message states that the variant was converted to a gene using dbSNP, and GDLPA shows the result for this gene. After submitting a variant, gene, or drug query, the GDLPA website displays a list of relevant drug-drug, gene-gene, and gene-drug interactions as top 10 suggestions for relevant related drugs and genes. Below this list is a dashboard of relevant resources presented as cards. Depending on the input, namely a variant, a gene, or a drug, the gene-drug interaction, gene-gene interaction, or drug-drug interactions are presented based on knowledge from literature conventions and mRNA co-expression. The mRNA co-expression is calculated from thousands of randomly selected RNA-seq samples processed with the ArchES4 pipeline. When a user submits a drug as a query term, GDLPA provides drug-drug and drug-gene interactions based on conventions and abstracts, and drug-drug interaction based on cosine distance between drug-induced length cell 1000 signatures calculated using the characteristic direction method. The resource card results are filtered based on the availability of hyperlinks for each resource. The results are sorted based on whether the resource is tagged as a primary source and also based on the number of previous clicks on each card. The structure of resource cards for genes and drugs is identical. The header of the card displays the resource affiliation, the resource website logo, and whether it has some affiliation with the CFDE. The body of each card has a screenshot of a landing page of a corresponding resource, a short description of the resource obtained from the resource page, and a hyperlink to the resource. Finally, the footer of the card displays a hyperlink to the landing page centered on knowledge about the specific gene or a specific drug. The gene and drug resource pages provide an overview of all genes and drug resources available from GDLPA. Each resource is displayed as a card but without a footer to the gene pages. Every resource card provides information about the center affiliation of the resource, whether the resource is affiliated with CFDE, the resource logo, a screenshot of the landing page from the resource, a short description of the resource, and a link to the resource. The cards are sorted based on the primary source tag and the number of times the card representing the resource was clicked. The Gene and Drug landing page aggregator website is a React application that uses Next.js. The site renders pages server-side and generates indexable and cacheable static pages. We utilize incremental static regenerated page caching to provide the benefits of static web pages for search engine optimization, and page caching properties to only recompute the pages after a period of time to ensure the information is kept up to date. GDLPA is not a typical knowledge aggregator for genes and drugs. Instead of storing and serving information from multiple sources, resources are queried on demand, and the user is directed to information about genes and drugs from the primary source. The manifest of resource lists all of the gene and drug-centered resources, including their descriptors, categorization tags, and callable functions to generate hyperlinks based on the gene or drug query. Manifests are resolved at query time based on the gene or drug information provided, filtered based on the availability of a hyperlink and sorted by whether the document is tagged as a primary source or not, and page ranked based on the number of clicks. To display the availability of genes and drugs across resources, we generated accessibility matrices. Genes and drugs are listed in the columns within these matrices, and resources are the rows. Bright colors represent the presence of a drug or a gene in a resource, while dark colors indicate absence. The Downloads tab provides access to the manifest of the gene and drug resources, as well as the matrices that describe specific gene and drug availability across the resources. The matrices were generated by querying all the gene and drug terms across each resource. These matrices are updated periodically. I would like to thank all the members of the lab for working on these projects and NIH Common Fund for support and funding.